Hello and welcome to our Bio 105 presentation. My name is Melanie and I will be going over the introduction. Zoe will be going over essays and methods. Next, Sonia will be discussing results. Anna Blake will be discussing our interpretation. And lastly, Zoe will be finishing us off with conclusion and future ideas. So what is tartrazine? Tartrazine is a synthetic yellow food dye used in food, cosmetic, and drugs. It is also known as yellow 5. As seen on the right, it is found in many of your favorite everyday foods such as Lucky Charms, Dorito, Gatorade, and even M&Ms. The bottom left shows the chemical symbol and makeup of tartrazine. Tartrazine is a highly debated food dye. This is because many researchers believe it can possibly cause harm to the body, as it's been linked to adverse reactions. In this publication, it poses the biological question of, does tartrazine display harmful effects on human health? It also asks the experimental hypothesis, which is just a statement that can be tested of, tartrazine causes a disruptive effect on vascular formation in zebrafish embryos and human cells, leading to purpuric spots, ADHD, and other adverse effects. So why should we care? Tartrazine is so widely used in everyday food and drug products, many that you could even be consuming, and it could be potentially causing harm to you or your loved ones.
This study utilized two organisms in its experiments: zebrafish, which are popular in biological research because of their clear skin and visible organs during development, and human endothelial cells, which form our blood vessels. The first study on zebrafish gave information on the overall toxicity of tartrazine. The embryos were raised in wells with or without tartrazine, and after 4 days, their physical shape, including any oddly formed parts, were analyzed. The scientists also recorded how many survived in each well. This experiment answers big questions like, is tartrazine toxic? How toxic may it be? And what areas of the zebrafish body are affected? The second zebrafish experiment used two different strains that were genetically engineered to have blood vessels with green or red fluorescent labels, the green one being EGFP and the red one being M. cherry. This allowed them to show up under specific microscopes and look like the example image I have on the lower left. The wild type strain, or the strain with no edited genes, was also used in the experiments, and I've included it here for clarity. The embryos were exposed to different concentrations of tartrazine and then analyzed under a fluorescence microscope, which showed how their blood vessel structures developed and gave the scientists information of tartrazine's effect on this development. Moving on to the human cells, I want to mention that some critical functions of cells are to grow and divide, form structures, and move to different places. These behaviors mark a healthily functioning cell. And in this case, properly working human endothelial cells are expected to proliferate, form tubular networks, and migrate. In 3 different experiments, the scientists exposed human cells to tartrazine and then measured their ability to do these behaviors. Cells were measured for their ability to grow and divide in the proliferation experiment, and for their ability to form important structures in the tubulogenesis experiment. In the migration experiment, cells were cultured to form a sheet that was then wounded or scratched with a micropipette tip in order to force the cells to go to either side of the scratch. The change in size of this wound from 0 to 6 hours post-wounding gave the scientists information about tartrazine's effect on cells' ability to spread out and migrate. Again, a very important behavior.
So now I'm going to go over the results of this paper. So first, after exposing embryos to differing amounts of tartrazine, scientists found that higher amounts of tartrazine did cause embryonic defects. So the graph on the left shows how these different defects changed levels as the concentration of tartrazine went up. And as you can see, the percentage of the surviving cells and embryos decreased as concentration began increasing. And another finding was that as tartrazine started reaching extremely high levels, survival rates of embryos decreased because these defects began occurring simultaneously, so the severity of their impact began increasing. Um, the next thing that they studied was the effect of tartrazine on blood vessel formation, and they found that tartrazine also had very negative impacts on blood vessel formation. So embryos got various doses of tartrazine, and at higher doses, blood vessel formation was abnormal. These vessels did not branch properly, and instead of developing distinctly, they started developing very clumped together, as can be seen in the picture on the left-hand side. And it was also found that vessels grew in a different shape than expected at higher doses of tartrazine, which can be seen in the pictures on the left. And in the last experiment, they used human cell assays to test whether the results obtained by using the zebrafish embryos were accurate. And they found that human cells were also not able to form proper blood vessels, and the branches from these blood vessels began forming at like abnormal levels, or they just stopped forming at higher doses. So these were the results of this paper.
So for the first part of this experiment, the scientists looked at the impact of tartrazine on zebra embryo survival. And we can see in the figure on the left that as the concentration of tartrazine increases, there is a sharp increase in malformation of the embryos and a sharp decrease in the survival of the embryos. And I agree with the scientists' interpretation of this graph because, as we can see, there's a drastic change in the survival and malformation as the concentration of tartrazine increases. And this shows that as more embryos are exposed to tartrazine, it impacts their health more. Another thing the scientists noticed was that tartrazine had different impacts on the embryos by showing different types of malformations or health impacts on the embryos. And the figure on the right shows that there are multiple different types of malformations that can occur. And the scientists concluded that some of the types of malformations decreased as concentration of tartrazine increased. And they concluded that this was because there could have been more severe malformations that are occurring in the embryos, but I don't necessarily agree with this because there were no further tests in the experiment to determine this. The next result of this experiment dealt with blood vessel formation in the zebrafish embryos, and in the image on the left, the scientists concluded that as the concentration of tartrazine increased from 0 grams per liter to 5 grams per liter to 10 grams per liter, the distinction of blood vessels became harder to identify. And as you can see in the bottom picture, it's very hard to distinguish the different blood vessels. The scientists also noticed that basket sizes in the zebrafish changed, and this is just a structure in the zebrafish that has to do with embryo development. And they noticed that in the embryo that was exposed to tartrazine, there's a smaller basket. And I agree with all of these interpretations by the scientists because it's very clear in the images that you can see the clumped blood vessels and smaller basket size. Lastly, the results showed that exposure to tartrazine can prohibit the human umbilical cells from proliferating or growing and dividing. And this is seen in these graphs as the concentration of tartrazine increases and proliferation in tube length and coverage decreases. And these images show that it's clear that increasing tartrazine exposure to cells can inhibit their growth.
In conclusion, this study discovered that tartrazine appears to have a harmful effect on blood vessel formation in both zebrafish and humans. The researchers suggested that tartrazine may interact with the bone morphogenic pathway, the CDC42 protein, or potentially multiple pathways. What this means is that tartrazine may interfere with cells' ability to tell themselves what they need to do in order to build blood vessels, but we're not sure how it's doing this yet. Additionally, if tartrazine harms blood vessel formation, it may affect how the brain develops, and it could be tied to the ADHD symptoms seen in children who are exposed to this food dye. Its ability to harm blood vessel formation may also be tied to the small blood pooling called purpuras seen in some allergic reactions to the dye. They don't know that there's a relationship like this yet, but it could exist. In the future, this study could lead to research that gives us a greater understanding of the risks of tartrazine in humans and result in FDA regulations being updated. And as a silver lining, tartrazine's ability to stop blood vessels from forming means that it could be an early form of an important medicine, such as one that can stop cancerous tumors from forming. This is the first study to suggest that tartrazine affects blood vessel development, and it is an exciting start to more discoveries about this chemical.